Hello everyone, myself Anamika Tiwari, Teaching Associate from Department of Biotechnology of Rama University. Today I am here to deliver the lecture on microbiology. My topic was historical perspective. Historical perspective, we were discussing about the Robert Koch. Robert Koch was a scientist. He was known as the father of bacteriology. He has discovered different type of bacterial infections like anthrax, cholera and rabies. He has also given the postulates about that bacteria, how the bacteria is infecting the healthy person from the unhealthy person. So his postulates were that he stated that the microorganism should be constantly associated with the lesions. Lesion is the wound if the patient there is the healthy person and healthy person. In healthy person there is the wound, lesion is the wound in which bacteria is present. So he isolated that bacteria from the unhealthy person and injected to the healthy person to observe the infection which unhealthy person is having to the healthy person. He has also said that if the bacteria is present in the healthy person, it will divide and get the infection which is present in the unhealthy person. His second postulate was it should be possible to isolate the organism in pure culture from the lesion of the disease. If we are taking the bacteria from any culture, we have to take the pure culture of that particular bacteria for the division and for the growth, to check the growth of the bacteria. Third postulate was that the same disease must be resolved when the isolated microorganism is inoculated into the suitable lab animal. He has also checked that bacteria is the infected person and taken the bacteria to the lab to test the same infection does any lab animal is getting or not. Fourth postulate was that it should be possible to re-isolate the organism in pure culture from the lesion produced in the experimental animals. He has checked that the particular unhealthy person is having the same infection, the particular healthy person is having the same infection, and the animal which he has isolated that bacteria is also having the same infection. He has checked the same level of infection and the bacterial condition which the bacteria is giving why the bacteria is dividing and giving the same infection or same disease to 3 of the living organisms. Our fourth scientist was Sir Ronald Ross. He has developed the life cycle of the malarial parasite in mosquito. He has also awarded the Nobel Prize for this experiment in 1902. Our fifth scientist was Joseph Lister. Joseph Lister is known as the father of antiseptic. He has observed that during the post-operative conditions, there were the equipment, surgical equipments and different type of equipments which are used in the operative condition. They were containing the different type of infection while they were delivered to the healthy person. So he has told that if the particular equipment was sterilized with the help of any disinfectant, it will that person will not get the infection through that surgical equipments. So he is known as the father of antiseptics because he has given the post-operative disinfectants condition. A sixth scientist is Edward Jenner. Edward Jenner is known as the father of immunology because he has developed the first vaccine which was smallpox. During his working condition, there was the farm of chickens and there was the farm of cows, the dairy of the cows. There were the workers of that dairy, and the same workers were getting the infection from the chicks and the cows. He has isolated that bacteria and checked the division and infections by how the bacteria is causing the infection to that person. He also taken that bacteria and injected to the boy to check that how the bacteria is causing the infections to the healthy person. He proved that the resistance to the smallpox can be induced by injecting cowpox materials vaccine from the diseases into the man. Our next scientist is John Tyndall. During 1820 to 1893, he explained that how the microbial growth can be observed. He has also introduced the Tyndallization process. In Tyndallization process, he explained that if we if we take the broth and boil it to the 100 degrees Celsius, the microbes present in that broth will be destroyed. He explained this process by giving the a certain chamber which was sterilized, and he has done this experiment again and again to check the microbial growth is being continued during the cooling condition and during the boiling condition. That's all for today. Thank you so much.